Hello students, now we will start with new topic, that is embryological evidences of evolution. Embryological evidence of evolution, it was proposed by Ernest Haeckel. It was disproved by Alfred Wallace. It was disapproved by Carl Ernst von Baer. Disapproved by Carl Ernst von Baer. What is meant by embryological evidences? Embryological evidences of evolution. Many embryos of different types of organisms show similar genetic relation among each other. Embryological evidence means many organisms during their embryonic development showed genetic relation with each other. So, this is just the meaning of embryological evidences of evolution. When we are studying in detail about this embryological evidences, first of all, we should recall what are the embryonic stages of different types of organisms. And also, what are different anatomical differences in the embryonic stages of different organisms. In case of embryonic development, we see the common feature, that is existence of germ layers, ectoderm, mesoderm and endoderm. Among all types of organisms we will see, but from which phylum do we see such type of germ layers? From the phylum Platyhelminthes to Chordata. The common feature is existence of 3 germ layers. What are those 3 germ layers? They are ectoderm, mesoderm and endoderm. Next, when we come to talk about only vertebrates, then what are the similar features among the embryos of organisms? When discussed about only vertebrates, we commonly have to consider from fishes to the chordates, to the mammals. We consider from fishes, that is species, to mammals. So here, what are the common structures that we are seeing? The common structures will be existence of tail, existence of gill slits, and also earbuds or ears, etc. Coming to tail. So this one coming to the tail part, which is present in embryonic stage. It is retained only up to the aves phylum. Later it got disappeared in the adult form of other organisms. It retained only up to aves phylum and disappeared in other adult forms which are in the hierarchy of the taxonomy other than the aves. After aves we can see the organisms like mammals, etc., hemichordata, and disappeared in next higher level adults. But in aves we can see existence of tail. Coming to gill slits. So these gill slits, these appeared in the fishes. So coming to gill slits. These gill slits in fishes formed into gills. There are some exceptions, but generally we are reading about this. The same gill slits in case of amphibians have been converted into lungs. Modified or replaced by lungs. The respiration by gills in case of fishes is termed as branchial respiration. The respiration by lungs is called as pulmonary respiration. Again I am repeating, the respiration in case of fishes by the gills is termed as branchial respiration. The respiration by lungs is termed as pulmonary respiration. But in cockroach, it is because of the spiracles and the tracheary structures. So there we call it as tracheary respiration in case of cockroaches. That is different concept. But coming to embryological evidences. So these are the basic information about the existence of embryos and different structures that are present in embryos from different levels. Here we are discussing from aves onwards. Next, there are some temporary embryonic evidences. That is presence of egg tooth in case of birds. So one topic is temporary embryonic evidences. This temporary embryonic evidence is studied with best example, that is egg tooth. Example is egg tooth. In the embryo of developing birds, during their embryonic stage, for suppose if you think that this is a bird, here in the embryonic stage there will be some structure. With the help of that small structure which is present at the tip of the beak-like structure, that is absent in case of adult birds. But when the embryo of the bird is getting hatched, at that time it is present as a white scar or a white thick layer. That is helpful for breaking the eggshell and coming the chick out of the egg. So this is called egg tooth. That is absent in adult birds. Since it is present only in the embryonic stage before hatching or even after hatching also, just for few hours or 1 or 2 days it will be present, and later it will be falling off from the chick. So since it is present only in the embryonic stage, we are giving a term like temporary embryonic evidence. So this is one best example. which is present only in case of birds. Present in only embryo of developing chick. Next, coming to the adult stage of the bird. Bird has retained few structures from the reptiles, that is laying of eggs, one character, and a small scaly like skin or layers at the bottom part of the legs. Some scaly skin is present, that is the characters of reptiles. Since those are the two characters of reptiles that are seen in case of adult birds also, we are calling birds as glorified reptiles. Another one, in birds, two characters. So there are many, we are taking two. Two characters. We can write here few characters. Few characters of reptiles are still existed in adult birds. What are those two or few characters? That is egg-laying nature and also scaly skin. Because of those two, birds are termed as glorified reptiles. So, what are few characters we have written? One is egg-laying nature. Next, clearly we can see the scaly type of skin at the leg area. Scaly skin. So, birds are also called as glorified reptiles. Later, these embryological evidences are also explained with a type of theory that is called as recapitulation theory. What is meant by recapitulation theory? General structures of any developed higher birds or any other hierarchy organisms, those are general structures are developed earlier than the special structures. So we will see meaning and some of examples of those words also. It was later explained with recapitulation theory. Recapitulation theory, it was given by von Baer, Carl Ernst von Baer. What is it telling? It is telling that general characters are developed earlier than special characters. So what are general characters of any living organism? Existence of brain, existence of a spinal cord, next existence of axial skeleton, next existence of aortic arches, etc. Then what are the special characters? Special characters are development of feathers, presence of feathers in birds, and hair in higher organisms, that is mammals. Hair in mammals. So this recapitulation theory was later modified in the light of evolution, means in the next course of evolution. So this was later modified in light of evolution. Light of evolution in the sense course of evolution. It means in the next evolutionary time. In the later evolution, the same recapitulation theory was given with a term called as embryonic parallelism. Embryonic parallelism. The same one, embryonic parallelism or recapitulation theory, was again given with another term by Ernest Haeckel, that is biogenetic law. Was explained or proposed by Ernest Haeckel with the term that is biogenetic law. So this Ernest Haeckel has given a statement that is ontogeny repeats phylogeny. Ontogeny repeats phylogeny. What does it mean? Here, the present existing organisms has developed some of the ancestral characters. The present embryonic development stages are the repeats of ancestral development stages. But this was not applicable for all types of embryos when we compare all the vertebrates to invertebrates or among vertebrates, because during a particular time of embryonic development, most of the structures are replaced by other types of stages. Here we can take example of this ontogeny repeats phylogeny as when mammalian embryos showed presence of gill slits, which later developed into lung-like structures, and tail. This tail is remained as tailbone or caudal bone in case of mammals. These developed into lungs in case of adult mammals. This one, tail, this remained as tailbone. We can also call as caudal bone. Next. This one, ontogeny repeats phylogeny. It was given in a book, that is generally in book written by Ernest Haeckel. That book is generally, the spelling may be quite different. Just try to remember the name. Morphology der Organisme. See, the spelling is quite different. Generally, morphology der Organismen. So, in this book, he has given this statement. Which person? Ernest Haeckel, regarding the biogenetic law or embryonic evidences. There are some types of structural developments. That is organ development. For suppose, in case of fishes, we can see two-chambered heart. In case of amphibians, we can see three-chambered heart. In case of reptiles, except in case of crocodiles and monsters, we can see 3.5 chambered heart. But exception is crocodile. In case of mammals, we see 4-chambered heart. Next, other embryological evidence is, even in case of adult also, the same thing is being repeated. So, organ development is different in different phylums. For example, if you take fishes, which are read under species, it is two-chambered. In case of amphibians, it is three-chambered. In case of reptiles, except crocodiles and dragons, we see 3.5 chambered. In case of adult mammals, we see 4-chambered. Even in crocodile and dragons also we see 4-chambered heart only. 4-chambered heart. Next, coming to paleontological evidences. Paleontological evidences. So, these evidences are based on fossils. What are fossils? Fossils are the remains or remnants of impressions of the skeletal system of the organism. So, it is based on fossils. Fossils means remains or remnants of impressions of skeletal system of organisms, because smooth muscles will be degraded and mixed with the soil. Age of an organism or age of a fossil can be decided by or tested by carbon dating technique. Age of a fossil is done by carbon dating technique. This is given by Libby. Next, father of paleontology is Leonardo da Vinci. Next, father of modern paleontology is Georges Cuvier. Georges Cuvier. Another term is there that is called as missing links. Usually links means about the organisms that have retained characters of two different phylums that we are reading under link organisms. But here missing links means already they are existed. After their means extinction, we are means comparing the links what they have retained during their lifetime. So missing links are the details about organisms that have been extincted by now, but when they are alive, they have been retained characters of two different phylums. Here example we can take Archaeopteryx. This has retained the characters of apes and also reptiles. We will see what are the characters that Archaeopteryx has been adopted during its lifetime. So, Archaeopteryx its reptilian characters are, first one, existence of teeth, means presence of teeth. It looks like big sized bird like that. Next, second character is presence of free caudal vertebrae, just like a tail, free moving tail, having free caudal vertebrae. Another one is it has claws, means having claws at the foot. Those are digit-like structures. Next, apes characters. In case of Archaeopteryx. Now, having a beak. But now Archaeopteryx is not existed. Having wings. So we can take some other examples also. Like having 3 fingers in toes. Next, having furcula. What is furcula? Furcula also called as wishbone. It is also termed in normal words like it is just like a means it is present just below the inner side of the sternum region. Wishbone, for suppose. If you think this is a bird, here will be the presence of the sternum, because birds will be having some special uh, swollen like structure. If you think that this is nothing but keel, this inner one which is written here is sternum. So we can write one more point, that is sternum has keel. So these are some of the characters that are adopted from the birds. Now, basing on these characters, we can assume that birds are evolved from reptiles. Means basing on all these characters in aves, but that is present in Archaeopteryx also. There are some other paleontological evidences that we will see. One is pteridosperm, which is the paleontological connecting link between ferns and gymnosperms. So example 1 is Archaeopteryx, we have seen. Now example 2 is pteridosperm. Which is connecting link between ferns. Ferns are studied under pteridophytes. And another one, gymnosperms. Now, we know that pteridophytes produce spores. Now, gymnosperms, they produce the naked seed. And they also exhibit secondary growth. These are the few sample examples of paleontological evidences. Another one we can take, example 3, as velociraptors. These are a type of dinosaurs. They are means adopted. They have been adopted the characters from reptiles and birds. Next, these velociraptors have wishbone. Just like that of birds. Next is biogeographical evidences. What we study under biogeographical evidences? How different types of organisms, means a particular group of organisms or different biodiversity in one particular area or in a geographical area, how there they showed different morphological changes in their body. So before studying about biogeographical, let's see general meaning of a biological evolution. A change in the properties of a group of organisms. We take one particular group. For example, fishes are there in oceans. So in an ocean, that is one specific area, geographical area, we see different types of fishes. But in course of evolution, there might be many changes in the shapes of the fishes that are still being studied. Likewise, if you take Darwin's finches, so there are different morphological beak shapes in the finches. What are finches? A small black bird-like structures which look like little bit some crows-like or sparrows-like birds. So this is just the meaning of biological evolution. How they are distributed in different geographical areas. So example we can take under Biogeographical Evidences of Evolution, Darwin's Finches. First, what is meant by finches? Finches means small sized black birds. Where are these? Means the special black birds or the finches. Where are these finches existed, or where are these birds that are studied by the Darwin existed? So Darwin's finches are existed in Galapagos Islands. Actually, this is not one single island. This is a combination of many islands. So, these Galapagos Islands are 22 small islands. Combinedly are called as Galapagos Islands. Now, what is the number of means different types of Darwin finches? These birds are of 20 varieties. So where are these Galapagos Islands located, or these 22 small islands located? These are located on west coast of South America. These 20 varieties of Darwin finches, they vary in two different characters. In two different characters or features. What are those two different features? One is beak shape. Other feature is size of the bird. At the same time, other person called as Dr. David Lack, this person has proposed that this variation in all these types of Darwin finches or the finches studied by Darwin is because of the food habits that have been adopted by these finches. Basing on the type of the grain, the shape of the beak has been changed. So normally we can write one point here. Darwin's finches have different food habits. That is responsible for different beak shapes. Later, Dr. David Lack proposed that different beak shapes are due to different types of grains that are taken as food by the finches, or different grain eating habits we can write. Now, this same example means basing on the types of food habits, the beak shapes are also means change. This can be explained under divergent evolution also. This Darwin finches examples are also explained in divergent evolution. So based on different habitats, many varieties in the organisms have been evolved. So this is because of the food habits. But there are some types of organisms which are living in different habitats. Means some live in polar, some live in tropical. Based on that also, different evolutions or different morphological changes has been adopted in the organisms. Based on habitats, different variations are seen in organisms. We can take examples like elephant. In case of African elephant, that is residing in one habitat. Indian elephant, it is residing in other habitat. The morphological appearance of Indian elephant and African elephant, it will be quite very much different. That we can consider under this divergent evolution, which is studied in this biogeographical changes or evidences. Next one. There are some discontinuous distribution of organisms. So another topic is discontinuous distribution of organisms. Means previously Earth is having one single geographical area, but during the course of evolution, the earth layers or the geographical parts have been dispersed into different parts, but only on the earth. For suppose, if earth is having one single geographical area, in course of evolution, land and water has been separated, and organisms which are present as one common area now have been dispersed into different lands. Okay, we can consider this non-circled areas as water and the circle areas as the distributed areas of different organisms. We can also frame this one as we know that most of the part of the geographical area on the earth is covered by water. Only little by animals or organisms. But all this is occupied by water. So organisms have been dispersed into different areas. Basing on that, we will see some examples. Plants like magnolia, tulips. They are found in China and eastern USA. Basically found in China and eastern USA. Next, elephants are there. Found only in India and Africa. This is naturally, we are talking about wild organisms, natural organisms. Later, different countries have taken these organisms and this got adopted as one of the part of those countries. That is different. But basically, how the organisms initially have been dispersed into different geographical areas for their survival. Next, another example we can take lungfishes. These are found initially later the different types of organisms have been migrated into different aquatic environments. Found in So under lungfishes we have different types. One type is Neoceratotus. This type of lungfish is seen in Australia. Another type of lungfish is Lepidosiren. This is seen in South America. Another type of lungfish is Protopterus. This is seen in Africa. So different organisms have been dispersed to different geographical areas. We can take another example, that is some organisms have been restricted only to one particular area. There we can consider the existence of marsupials. For suppose you take kangaroos, they are still permitted to one geographical area, that is Australia. Next, restricted distribution. Here we can take examples like marsupials and also egg-laying mammals like echidna. Those names of organisms are kangaroos, and this one we can take echidna. Next, so we have seen one example under divergent evolution. Now coming to convergent evolution. Some of the examples here. Next is convergent evolution. Here, different organisms are having different origin in nature, but their functions are same. Those are the organs which are doing such type of characteristic features are also termed as analogous organs. Origin of organisms are different, and their body organs are also different, but functions are same. Here, we can consider these organs as analogous organs. Analogous organs. Now, basing on this, we have few examples like placental mammals, next, and normal mammals. Then what is difference? Placental mammals, they directly give rise to the infant, but normally there are some special types of mammals, those give rise to larvae. They give rise to infants. Here we can take examples like moles. Here we can take, they give rise to larvae. Here we can take examples like marsupial moles. These look like a mouse-like structures or rat-like structures. So under this one, moles. These are also other type of moles, but these are called as fossorial mouse-like structures. These moles are means which live in the burrows are called as fossorial organisms. Fossorial mammal mouse. Fossorial mouses. Actually, they are color blind. This one. These are color blind. This is about biogeographical evidences of evolution. Next is biochemical evidences of evolution. So, these biochemical evidences of evolution are responsible for cytological changes in the organism and also physiological changes in the organism. Cytological changes and physiological changes in organisms. Now here we can take many examples. Already we have read what is meant by biochemical structures or substances. Nothing but all types of organic substances like proteins, carbohydrates, the enzymes that they are secreted, the lipids that are present, the nucleic acids. All these have brought many variations in organisms. Here we can read some of the examples like enzymes like amylase and trypsin. These both are considered as universal enzymes. Next, these are seen from the phylum Sponges to mammals. So we are giving a term as universal enzymes are seen from even amoeba is also having amylase for its digestion of the food from sponges to mammals. We can also write amoeba-like organisms and amoebas. Other example we can take is about hormones. For suppose, if thyroxine is injected in tadpoles, then the hormone stimulates metamorphosis. If this thyroxine hormone is deleted from tadpole larvae, then it will not show the molting or the shedding stages that are present in its lifetime. When thyroxine is injected in tadpoles, larvae of frogs, the metamorphosis means morphosis, the shedding or molting stages seen is stimulated. Next one, other examples is cytochrome B6 and cytochrome C, etc. Other examples are blood and lymph, seen in all vertebrates. Next, blood proteins These are also seen mostly in almost all types of vertebrates. Seen in all vertebrates. Next, here we can see some of the structures like hematin crystals which are present in chimpanzees, apes, etc. But those are similar to the blood proteins of human beings. Next, blood proteins in apes and humans are similar with blood proteins of herbivores. We can take of herbivores, means the blood proteins of carnivores are similar to that of herbivores also. Next, another example we can take like blood groups. AB blood group is found in humans and apes but not seen in monkeys. AB blood group. This is AB blood group seen in humans and apes but not in monkeys. Next, some of the excretory substances. If the organism is purely aquatic, then it will be ammonotelic type of organisms, means it will be excreting ammonia. If water is less, then that type of ammonotelic nature will be converted into ureotelic or uricotelic. So that examples we can write. Excretory substances are mostly seen in aquatics and that is ammonotelic nature. In case of xerophytes, means in case of terrestrial organisms, terrestrials will be mostly ureotelic or uricotelic, means when water scarcity is present, because for excretion of 1 gram of ammonia we need 500 ml of water. So this is about some of the examples of biochemical evidences of evolution which are responsible for the cytological and physiological changes in the organisms in the course of evolution.